Amphibian metamorphosis is under neuroendocrine control and it involves neurosecretory cells in the brain, that is, the hypothalamus, and the two endocrine glands, that is, the pituitary and thyroid. The trigger to metamorphosis may be an environmental signal affecting the larval brain through the nervous system, or there may be an endogenous clock in the hypothalamus. In a way, hypothalamus integrates the information received from the body with environmental information. We can say that during metamorphosis, developmental processes are reactivated by certain hormones. Thus, metamorphosis is often a time of dramatic developmental changes affecting the entire organism. This is a picture of a tadpole. We know that tadpole lives in an aquatic environment, and with the passage of time it has to change and prepare itself from an aquatic being to a terrestrial animal. In case of amphibia, where we study two class, one urodels, example salamander, and second anurans, example frog. In these two forms, we have seen that how from being an aquatic animal they become terrestrial. In case of salamander, for reproduction they need to go back to water. So in urodels, example salamander, there are morphological, physiological changes, or we can say metamorphosis occur two times, whereas in anurans, example frog, from the eggs tadpole hatches. They have all the necessary equipments to live successfully in water, and after passage of time these structures regress. New structures arise. Regression of old structure and new structure origination both are caused due to hormones. The structures like external gills, tail, aquatic skin structure, alimentary canal, they all regress, they all transform, change from aquatic type to terrestrial type. So we can say that in them, in urodels and anurans, regressive as well as constructive changes occur. As I have already mentioned, means of locomotion changes. In frog, the pedal tail recedes, cartilaginous skull is replaced, horny teeth that are present in tadpoles are replaced, tongue muscles develop. Since tadpoles are herbivorous, their alimentary canal structure is such as to eat and digest the herbs, the grass, or the vegetation which is around the animal in aquatic environment. So the large intestine has to be long. in case of tadpole, so that the herbs and the grass etc. are digested and absorbed rightfully. Whereas we know that adult frogs are carnivorous. So when the metamorphosis occur, there has to be changes in their intestinal composition. Hence, the large intestine shortens. Since animal now does not live in water, the gills regresses, lungs develop, sensory appendages also changes as the lateral system of tadpole degenerates, and eyes and ears, they undergo differentiation. Middle ear and eyelid develops in frog. Nervous system also undergoes dramatic changes. In anurine metamorphosis, the movement of eyes forward forms their originally lateral position. In some larvae, neurons in tadpoles' jaw switch their allegiance from larval muscle to newly formed adult muscle. Other neurons innervating the tongue first form synapses during metamorphosis. Along with the morphological and physical internal changes, biochemical changes also occur, like the pigment in tadpoles, which is porphyropsin, changes to rhodopsin. Hemoglobin also changes. The adult hemoglobin binds to oxygen more slowly than as it was in tadpole. The enzymes in liver also changes, because tadpole, as it is living in water, the nitrogenous waste is excreted in form of ammonia. But when its life is changed from tadpole to frog, then the nitrogenous waste is not excreted out as ammonia, it is excreted out as urea. So the liver enzymes changes for the conversion of nitrogenous waste into urea. So new enzymes like ornithine carboxylyl transferase, arginosuccinate synthetase, arginosuccinate lyase, arginase, etc. These enzymes, they are formed. This is the summary of metamorphic changes that occurs in frog. Locomotory in larva, it is done by the tail, while in adult it is done by the hind limbs and the forelimbs. Respiratory is done by gills, skin and lungs. In tadpoles, larval hemoglobin is present in tadpole, whereas in adult, the respiratory system is formed by skin, lungs and adult hemoglobin. The gills regresses. Circulatory system also sees change. Carotid arch system, systemic arch and cardinal veins etc. develops. From nutrition, from herbivorous lifestyle, it becomes a carnivorous. So the long spiral gut also regresses. It gets shortened. Certain enzymes are which is not found in the tadpole. Now they are found in the adult animal. Nictitating membrane, which is absent in tadpole, develops in adult. There is a development of ocular muscles, presence of nictitating membrane in adult. Rhodopsin is the eye pigment present in the adult, whereas in larval form porphyropsin is the eye pigment. The lateral line system, it degenerates in adult, which is present in the tadpole. Excretion is in form of ammonia in larvae, whereas excretion is mainly in form of urea in adult. So likewise, these changes are seen in the frog. The neurosecretory cells in hypothalamus are stimulated to produce thyroid releasing factor, which stimulates the anterior pituitary gland to secrete prolactin. The thyroid gland secretes the thyroid hormone. Increase in thyroid hormone trips the orderly sequence of tissue change that transforms the tadpole larva into frog.